I coordinate our biodiversity initiative on campus, and I work with a variety of new students each semester, and we focus on biodiversity initiatives. I have my undergraduate degree from UCLA in biology and environmental engineering, and I have my graduate degree in evolutionary ecology. And I've been here at College of the Canyons for 15 years. And it's been great. I've gotten a tremendous amount of support from many people here to pursue this really important topic of biodiversity. I coordinate many courses in our biological and environmental sciences division, and it's just great to be here.
Great, thank you.
So let me just start with this presentation. As I mentioned, we do have this really incredible biodiversity initiative on campus. And the goal of this initiative is to provide students an opportunity to work on projects and research that focus on biodiversity with the ultimate intention to increase biodiversity. It's an initiative that any student can participate in. And it really is neat because we use our campus as a living lab. So students get to get into what's going on in our campus. They get to work with faculty and staff and various community partners. So this is just some examples of some signage that students have made to bring awareness to a lot of the beautiful natives we have on our campus. This is a shed that students just transformed into a biodiversity storage shed. So the reason we feel this initiative is so important and our campus is prioritizing it is because biodiversity loss is happening rapidly. The World Wildlife Fund just reported that on average 69% of global species are in decline within the last 50 years. Within the last 50 years, North America has lost nearly 30% of their birds. And the World Economic Forum just came out with their global risk factor report, and they list biodiversity loss as the third most concerning risk factor. And when you look at the first two, climate action failure and extreme weather, of course, those two are related and they impact biodiversity loss tremendously. So we're focused on being part of the solution. And as Jason previously said, our guiding principle are the United Nations Sustainability Goals. And although 14 and 15 relate directly to our initiative, I think that every single one of these relates to biodiversity. So today you get the opportunity to meet 5 of our team members. These are students who are working on various projects. I'm going to hand the presentation over to them in succession. They will each introduce themselves and then give you a very brief description of what they're working on. So we will start with Alex.
Hi, my name is Alice Anderson Lau, and I'm in Environmental Studies, Water Systems Tech, and Geography, and I'm going into urban planning. I wrote a proposal to COC facilities to replace non-functional turf on the Valencia campus with a native habitat with the main goal of water conservation and lowering the impact of urbanization. Next slide. In May of 2022, the State Water Resources Control Board announced that the watering of non-functional turf would be banned. This defined it as solely ornamental and not regularly used for human recreational purposes. I clarified that functional turf is a place of say and purpose that requires at least 5 minutes of activity per group, with at least 3 series found in my 119 total observations, an example which is going to be found in the bottom right graph. In my top right graph, it shows that all the functional turf I found on campus with a numbered section of turf in bold. You can see that turf number 38 is the most used, and it makes up 60% of all of my observations. Turfs number 52, 11, and 5, which I proposed for a native habitat, are not found, which makes them non-functional. Number 52 is chosen due to its seclusion and proximity to a native walkthrough. Number 11 for the closeness to the community garden and hillside, and number 5 for its large location and the fact that it can be far from any other nature. The Valencia campus should be accessible at every corner. Next slide. Non-functional turf has to be replaced, so why not make it a native habitat? Well, native habitats increase biodiversity with pollinators, reptiles, and other species, which improves the mental health of students. This allows for learning opportunities as well. As you can see in the bottom right table, the red highlighted region shows that native plants receive an additional 25% of rebates from SCV water. More savings occur since native plants do not need any tech, which saves water and electrical costs. They will also naturally adapt to the soil and climate, and there's also pride at COC knowing that we can help and create and conserve a native sanctuary. Next slide. So why shouldn't we have non-native drought-tolerant plants in artificial turf? Well, drought-tolerant non-natives are often invasive and require higher maintenance since they are susceptible to local disease, predators, and the climate. Artificial turf is an even worse option since it has the heat island effect, runoff, and pollution, and a large carbon footprint. Now, what opportunities it opened is thanks to this program, I want to pursue the environmental impact reports, and I received an internship with the LA Sanitation for water and treatment observations. I implore any students interested to get involved with the environment on their campus. Thank you.
And I'd just like to quickly mention that Ellie is on schedule to present this proposal to the Vice President of Facilities next week. And we hope to implement at least some of her suggestions very soon. So next we have Morgan Phillips. Morgan?
Hi, my name is Morgan Phillips. I'm a biodiversity intern at COC where I study biology and hope to transfer into marine biology. The driving question behind my project is, does runoff contribute to high levels of bacteria in the ocean? And the goal is to establish a correlation between high precipitation levels, developed land, and high Enterococcus levels. And Enterococcus is a commonly used fecal indicator bacteria to measure bacteria levels in the ocean. For example, Santa Monica Pier has high bacterial levels, and it is due to pigeon fecal matter surrounding the pier. As you can see in the top image, it's shown there are holes in the fence allowing the birds to get in and poop. Sometimes the birds get stuck and die, as you can see in the bottom image, making it problematic for that reason as well. And then finally, the process of my project involves studying 25 drainage sites along the coast of California. I drew transect lines stretching half a mile long, projecting the direction of runoff flow. Next, I categorized the surroundings nearby each line and used this to establish my predicted bacterial contribution level using calculated mean values. Next slide. This is an example of one of my transect lines, a drainage site at Solana Beach with a calculated mean value of 5.536. There are 3 main divisions on this line. The blue section is showing sand, a low bacterial contributor. Yellow sections are restaurants, a moderate contributor, and pink is high density housing, a high contributor. This means an average value of around 5 is a fair summary of the low, moderate, and high bacterial contributors present on this line. Next slide. This project presented me with many opportunities, particularly through outreach. I met with Luke Ginger, a water quality specialist with Heal the Bay, where he works to produce the Heal the Bay report card every year, showing the bacterial levels of local beaches. Additionally, he provided me the storm drain beach list, which I used to select each of my drainage sites. He also introduced the USGS water data site and California Water Board's public records, where anyone can access the various bacterial levels at the beach. The outreach aspect of this internship has already presented me with another internship opportunity, Heal the Bay's river data collection position. Completing my project this semester heavily relied on scientific research as I learned to accurately interpret scientific results and realize the importance of persistence when ideas fell through. This project certainly had its difficulties, but it's made me proud of what I've accomplished. Thank you.
Thank you. And next we have Xavier Adams to present his project. Xavier?
Hello, everyone. My name is Xavier Adams, and I'm an intern for the College of the Canyons Biodiversity Initiative for the fall of 2022. In reviewing the work that I've been indulging with in the past few months, I will first summarize the results from my research, and secondly, we'll report the findings of my project. Beginning with my research, given that I am a political science major, I wanted to reconcile political science and biodiversity. Economic inequality is a topic that's dear to my heart, and eventually I decided to examine the link between economic inequality and biodiversity loss. I then gathered 3 scientific articles with the common aim: can economic inequality explain the varying distributions of biodiversity? After mulling over articles, it was revealed that high levels of economic inequality had, for the most part, demonstrated a significant predictive power for biodiversity loss among both countries and US states. Next slide, please. In two of my articles, the authors use birds as proxies for biodiversity. In recognizing this for my project, I sought to identify any changes of the College of the Canyons of Valencia campus bird species population, thereby measuring any change in its biodiversity. After several visits to each bird monitoring location found on the right, I then compared my fall 2022 data with prior fall 2018 data. Similarly, I compared prior spring 2022 data with spring 2018 data. Next slide, please. These graphs display the four-letter code of each observed bird species along the x-axis. For example, YEWA equates to yellow-winged warbler. For each species, the graph illustrates what proportion of the campus population they constituted in 2022 and 2018, being the y-axis. There are large discrepancies, as you can see here. To point out a couple, no band-tailed pigeons were identified in the spring of 2018. Additionally, the common ravens nearly doubled their proportion between the fall seasons. Next slide, please. In addition to my bird observations on campus, me and Ali, a fellow intern here, and Professor Chari made an effort to clean out the birdhouses around campus for the spring. Using the map to the right, detailing what type of birdhouses are on campus and its location, we cleaned out the oak titmouse and the bluebird birdhouses. We additionally added 4 new bluebird birdhouses on campus. Overall, the Biodiversity Initiative has served as a rich and rewarding learning experience, allowing me to not only refine my research skills, but my data analysis skills as well. I certainly hope to work to sustain biodiversity efforts in the future, even as a political science major. Thank you.
Thank you. And next we have Joshua Muck. Joshua?
Hello, everyone. My name is Joshua Mock, and I'm an AOC-COC student, and my research and internship was on bees. Through my internship with College of the Canyons, I was able to learn and dive deep into California native bees. So my project first started with analyzing and choosing 12 key species that are native to Santa Clarita Valley. These species will be representatives for the respective genera upon their active season. From the research papers that I consulted, I chose 6 plants that would be able to support these key pollinators throughout their respective season. I checked with the Santa Clarita Valley water database on each plant to determine whether they're compatible and non-harmful to our area. So you might ask, why is this important? We need to provide flora resources for these key native pollinators to support them for the entire bee season. We cannot just import or use billions of non-native honeybees to pollinate all these plants, for they're incapable and ineffective. Next slide, please. Many native bee species only have a single generation per year, while others are multi-generational, and some species are social and have longer flight season, being active throughout the whole year. With that being the case, I narrowed down 6 key plants that can support these native bees throughout their respective season. And as you can see, the plants that I've gathered support these native bees throughout the whole year. Next slide. I then moved on to analyzing and identifying different specimens from the Tejon Ranch area. Professor Chari provided me with over 30 specimens, which I had to determine whether they were truly bees or other types of insects. I had to pin and take pictures of each of these native bees, one dorsal, one lateral, ventral, and a picture of their face and wing. These pictures allow me to use a guide to possibly identify them. A part of the bee I want to highlight is the wings. Depending on how many marginal cells and the proportion of the jugal lobe, we can narrow down different genera of bees, almost like a fingerprint. Native bees can come in all shapes, color, and sizes, some being small as the tip of a ballpoint pen, while others are the size of quarters. Moving forward, we plan on continuing to collaborate with the Tejon Ranch Conservation and Antelope Valley College. An observation was noted that there was no overlap in species between College of the Canyons and Tejon Ranch Conservation. However, there was an overlap for Antelope Valley College for both locations. We plan on gathering samples along the path from College of the Canyons to Tejon Ranch to hopefully observe any patterns for this phenomenon.
Thank you. And lastly, we have Laurel. Laurel?
Hi, everyone. My name is Laurel DeSantis. I'm a biodiversity intern as well as a biology major. So my project focuses on biodiversity outreach. I wanted to spread awareness of the loss of biodiversity to more people, including like younger students, and so educate them on how to slow and prevent the loss of further loss of biodiversity, and then give them the tools and the resources they need to educate themselves beyond me. Next slide, please. So I met with 6 groups overall. I met with 3 high schools and 3 Girl Scout troops. The 3 Girl Scout troops I met with, the ages ranged from 2nd grade to 11th grade. And then the schools I met with Saugus High School, where I taught 6 biology classes, West Creek Elementary School, and Sulphur Springs Elementary School. So over the course of my project, I met with approximately 260 students. Next slide, thank you. So with each group, I had 2 different PowerPoints that I would show them. One was for younger students, like middle school and younger, and then one was for older students, so above middle school. And I pretty much just taught them like the who, what, where, when, why, and how's of biodiversity, and I answered any questions that they had. And to teach them what they can do to help prevent biodiversity loss is I gave them little seed packets. So these seed packets have native seeds in them, and they also have the care instructions on them. And I just taught them how to care for them, how to water them, where to plant them, that kind of stuff. And then with the students as well is we made these paper boxes, which is you can see on the top, I have one too. I made them with the students and they're made out of recycled newspaper that I collected from my family and from our neighbors. And with these, they can just go home, make more and plant their seeds in them, and then they can germinate in here. And then when they're ready, they can put them in the ground and they can decompose in the ground. Next slide. So this is the seed list of everything that was in these little packets. I made sure that they're all native to Southern California, but then I also cross-referenced with SCV water to make sure that they're compatible in Santa Clarita. And then all these seeds, I had to make sure that they can all grow together. Like they had similar light needs, water needs, soil needs, all that kind of stuff. And then next slide. And so these are some photos that I took while I was with the students. I don't have a lot with like them themselves because of they're younger and, you know, privacy stuff. So the top 2 are classrooms that I visited, and then the rest are all Girl Scout troops.
So that's our presentation, and thank you very much.